news and, oh no, there's a new variant of COVID-19. How did that happen? And what does that mean for us? Before we get to new variants, first we need to understand DNA. DNA consists of three parts: a phosphate ion, a deoxyribose sugar, and a nitrogenous base. These make up a nucleotide. There are 4 nitrogenous bases: adenine, guanine, thymine, and cytosine. In RNA, thymine is replaced with uracil. When you think of copying DNA, I'm sure your first thought is replication.
Yeah, right.
But transcription is so much more important. Transcription produces mRNA, which carries the information for creating proteins. Basically, RNA polymerase II, an enzyme, binds with DNA with the help of transcription factors. The transcription initiation complex then moves down the DNA and copies the DNA into RNA.
But what about those proteins?
The mRNA will bind to a ribosome, an organelle specifically designed for creating proteins, and begin translation. During translation, tRNA will bind with mRNA,
A lot of RNAs, I know.
to create a chain of amino acids. After translation, the amino acids fold themselves into a protein. These proteins can be either used inside the cell or exported by the endoplasmic reticulum.
Okay. But what do these processes have to do with virus strains?
Viruses are just genetic material, either DNA or RNA, surrounded by a capsid or a protein shell. They don't have the ability to replicate the genes themselves. Instead, they enter a host cell and force the cell to replicate the virus's genes. The host range is often extremely limited, sometimes to only a specific type of cell. The newly replicated viruses then destroy the cell to escape and continue infecting the host. Considering how complicated the gene transcription process is, it makes sense that there are often errors. Most are repaired by nucleases, but some are missed. This is how we get mutations. There are 2 types of mutations. A point mutation only affects one set of incorrectly paired nucleotides. Depending on the location of the nucleotide, this might not affect the result, or it could code for the wrong amino acid. The other type is a frameshift mutation. An insertion or deletion occurs that causes all the following codons to shift by one nucleotide. This occurred in the alpha COVID-19 strain. Because of the process of translation, mutations affect not only the genetic material, but also any proteins formed, including enzymes and the capsid. Changes to the capsid affect the host range for a virus. This is why when you hear about new strains, they often come with new symptoms. Or, with mutated enzymes, the virus reproduces more quickly and becomes more infectious. Even worse for us, the virus can mutate so the vaccines are no longer effective. This sounds terrible! So what can we do? Well, the short answer is nothing. We can't unmutate a virus to replace a more contagious variant. However, we can prevent compounding mutations. Because new strains are caused by more replication of viral genetic materials, it is important to continue to wear your mask and get vaccinated to prevent the spread of COVID-19.